So, a very warm welcome to all of you guys. Good morning, good afternoon, good evening, good night, depending on the time when you are watching this video. So, in this video tutorial, we will going to talk about the types of vectors. Types of vectors. So, the first thing that we will going to talk about is the cloning vectors. Cloning vectors. So, this is the most basic type of vectors we are using from the beginning of the recombinant DNA technology for our cloning procedure, right? Although we are not using cloning vectors nowadays because we have now PCRs, right? This PCR will give you million copies of your gene of interest within 1.5 hours. So why you use cloning vectors and wait for 3 days, right? So that is why nowadays cloning vector is a kind of old term. Still, so cloning vector, as its name suggests, it is a Xerox machine of your gene of interest, right? So that is the idea. So what are the components of cloning vectors? So cloning vectors have origin of replication, along with it should have a marker gene, right, and MCS, right, that is your multiple cloning site. Okay, so now after getting a huge success on cloning with the help of these cloning vectors, now scientists try to think about this, that okay, we have sorted our problem in the amplification of gene of interest. Okay, now we have PCR, so the problem is totally solved, right? So now why should not we try to get the protein that our gene of interest encodes, right? And from that thought, they developed a new type of vector named expression vector. Okay, expression means it will express the gene of interest it carries. Okay, so it is very clear that with one origin of replication, MCS and marker gene, we cannot express our gene of interest, right? We should need some more things, right? Obviously we need origin of replication, MCS and marker. Without this we cannot think about cloning, and if replication is not possible, so transcription and translation is of no meaning, right? Along with that, along with this tree, we should need some extra things, okay, that will help in our expression. So now we will going to talk about the components of a expression vector. So this is a drawing of expression vector. So along with origin of replication, as you can see here, origin of replication, selectable marker and MCS are present. Along with this tree, it contains a promoter, a operator, a DNA sequence which on transcription gives you ribosomal binding site, a transcription initiation site before ribosomal binding site, because remember it is in the DNA form, right? So for binding of RNA, it should be transcribed into RNA. That is why we have to incorporate ribosomal binding site after transcription initiation. Okay, and then after ribosomal binding site there will be MCS, that is the insertion point for your gene of interest. Okay, along with that there should be transcription terminators, right? Then another important thing, we should need some genes which will encode repressors that will bind over operator and regulate promoter. So that is all the ingredients that we need for the expression of a gene of interest. So when we will deliver this gene of interest containing vector inside a host, what will happen? RNA polymerase will bind over promoter. So firstly, let's try to talk about this from the beginning. First of all, from the origin of replication, with the help of DNA polymerase, replication will occur, right? Then after that, RNA polymerase will bind over promoter and start transcription. Okay, once transcription is completed, Bacterial ribosome will bind over the ribosomal binding site and starts translation, and as a result we will get protein. Okay, so that is the concept of expression vector. Now there is a situation that I am going to explain that is very important, not only for your university exams but also for competitive exams. Okay, so let's consider a situation. We are using prokaryotic expression vector containing prokaryotic origin of replication, prokaryotic promoter, prokaryotic operator. Okay, now interestingly we are using a eukaryotic gene of interest. Okay, so and we are transferring them inside a prokaryotic host. Okay, so what will be the consequences? That is the important question that you should understand. So there can be three fates of the gene of interest that I am going to talk about. Firstly, let's say origin of replication works properly, we get the replicated part. That is the first thing you should understand. Now, so let's now try to think about this. Promoter also works properly, operator also works properly, and we get RNA from the gene of interest. Okay, now ribosome will bind over here and also translate. Okay, now I want to told you that in case of eukaryotic host, after translation there is a special type of step named post translational modification. Okay, now that post translational modification will further oligomerize and further fold your protein. Okay, gives its activity, right? So now this thing is not usually observed in case of your prokaryotic host. Okay, now under such circumstances, can your prokaryotic host will try to help you? No, that is not, because you are not giving him salary for that, and without salary in this universe no organism try to help you. That is the reason your protein will not properly oligomerize or fold it. Okay, and as a result you will get a non-functional protein in some cases. Okay, I am not telling that you will get this every time, because let's consider some proteins that is not properly folded but still they are active. Okay, they are not having too much amount of folding. Okay, under such circumstances you can easily done the expression and you will get the protein. Okay, so that is the first fate, that the protein that we will get will not overfolded, and secondly it is not oligomerized. Now let's talk about the second fate. Second fate is, let's say origin of replication works properly, promoter works properly, operator works properly. Now in times of gene of interest it gets RNA, right? Now we all know that in case of eukaryote there are two type of RNA. One is exon, that is the coding genes, another one is intron, that is the non-coding genes. Okay, and after transcription there is a process in eukaryotes named intron splicing, or the splicing process that will splice the non-coding gene. Okay, and gives you only the coding gene that ultimately gives you protein, right? So these things is usually absent in case of prokaryotes, because we all know in case of prokaryotes there is no place for introns, right? That is the reason why the gene of interest having introns cannot be expressed in prokaryotes. Okay, so and that is also the reason why you can get a question in your university exam that, let's say, they will tell you why it is preferred to insert a cDNA of eukaryotic gene inside the MCS when you are planning to transfer your vector inside a prokaryotic host. Because remember, cDNA is coming from RNA, mainly mRNA, right? So a DNA which is coming from mRNA should be devoid of non-coding genes, right? That is the reason why we are generally prefer cDNA when we are willing to get expression and we are using prokaryotic expression vector and prokaryotic host. We have talked about an example earlier, that is GCSF production, where we have talked about, try to remember this, we will use cDNA, right? Not only that, try to, if you go back in my application of recombinant DNA technology video, there you can find out that in case of human growth hormone we have used cDNA, right? And we get a good expression inside a E. coli, right? That is the reason, because cDNA is devoid of non-coding genes. So that is the fate number 2. Okay, and fate number 3 is, in case of inside a prokaryotic host, you may not get the appropriate initiation factors that is required to start a eukaryotic promoter. Okay, that situation I am so sorry for that. This may not fit with the situation that I have considered here. The thing that I am telling you, that is, suppose you have taken a eukaryotic promoter, eukaryotic operator, eukaryotic gene, and you are sending it inside a prokaryotic host, then what will be the consequences? Under such circumstances, the initiation factors that your prokaryotic host provide cannot recognize promoters, and as a result transcription will not start. That is the idea, but it is not required, or you can also remember about this, that is no issue. Okay, so that is the thing that I am wanting to explain you. So you may get such type of question, please be ready for that. Okay, now after clearing our concept on expression vector, now scientists face a new challenge. What is the kind of challenge? So they sometimes try to use a prokaryotic origin of replication, okay, and this delivering it inside eukaryotic host, because their gene of interest, let's say, was eukaryote. Okay, under such circumstances, sometimes they find out that replication or cloning is not occurred. Okay, similar in case of, let's say, they use eukaryotic origin of replication and transfer this inside a prokaryotic host, and they not receive any kind of cloning. That means scientists try now observe that we have to choose our vector depending on, we have to choose our host depending on what is the origin of replication in our vector. Okay, or the vice versa is also possible, that we have to choose our origin of replication depending on what is our host, right? So scientists try to think about this, can we solve this problem, because this is a type of burden, right? Why we are going to try to think about this? We will going to use a vector and we will send him, that is all. So why we need to think about the what is the host, what is origin of replication? Can we not derive a solution for that? So research again begins, and after several research they get a idea that how it should be if we add origin of replication for prokaryote and a origin of replication for eukaryote, how it should be. And as a result, what will happen, because as it is a circular DNA, it will go for bidirectional replication. So as a result, if there is a gene of interest insertion site present and we will try to incorporate them inside a eukaryotic host, this eukaryotic origin of replication will function. Similarly, if we try to insert them inside a prokaryotic host, this prokaryotic origin of replication will start to function. So that is the problem solving approach, right? So we have constructed a vector that have the ability to shuttle our gene of interest back and forth between two origin of replication coming from both eukaryote and prokaryote. That means this vector can transfer our gene of interest in both the prokaryote and both the eukaryote. That will be the exact word. The previous thing I have said is not correct. We are shuttling our gene of interest back and forth between two hosts. Okay, one is your prokaryote, another one is eukaryote, and we are getting expression of our gene of interest in both the cases. Okay, so that's amazing, right? And as this vector shuttling gene of interest in between two hosts, so that is why they given this vector name called shuttle vector. So that is the concept of shuttle vector. Now there is a question. So okay, we have developed shuttle vector. This is a very good news for us, right? So now you may ask that yes, we have constructed shuttle vector in terms of origin of replication. So why we cannot add two promoter for both the eukaryotic host and prokaryotic host? Why we cannot do this? Because remember it, yes, there is a no problem of adding origin of replication for prokaryote and origin of replication for eukaryote. Okay, but in terms of promoter, remember that for every gene of interest there should be only one promoter. Okay, you can use only one promoter for the transcription of your gene of interest. Okay, that is the reason why we may add a promoter for prokaryote or we may add a promoter for eukaryote. That is why the concept of shuttle vector is not too much popular in terms of expression, but for cloning it gets huge success. Okay, so that is the concept of shuttle vector. So shuttle vector things are mostly performed when we need to study our genes expression in yeast. So in such cases, let's say we have constructed the plasmid in E. coli, we have manipulated the plasmid in E. coli. Now for study we are transferring it inside the yeast. In such cases we mainly use shuttle vector. Remember, for the study of genes inside the yeast, the gene of interest that we are using, 90% cases they are shuttle vectors. Okay, in case of yeast we may add a origin of replication extra for the maintenance of the gene of interest inside the eukaryote, and in some cases those gene of interest can integrate with the chromosome also. So that is the concept of the shuttle vector. I hope you have understood this very carefully, right? So first of all, the cloning vectors, which will give you the Xerox copy of your gene of interest. Then the expression vector, we will give you the protein from your gene of interest. Then we have talked about the shuttle vector that solves the problem in terms of cloning, that we can use any type of vector without bothering about what type of host we have, right? So that is the concept of shuttle vector. So I hope this video will be helpful in your preparations, depending on what type of preparation you are doing. And so thank you for listening to this class, and bye bye.